Hello and welcome to my presentation about DNA-based nanonetworks, or how to compute arbitrary functions at the nanoscale. My name is Florian Lau, and today I am going to present our results to you. I want to begin with a short size comparison at the nanoscale, since we use DNA as a material and not everyone might be familiar with it. What we see here is a size scale from 10 to the power of minus 1 nanometers to 10 to the power of 8 nanometers, which is the size of a tennis ball. The interesting interval lies between 1 and 10 to the power of 2 nanometers, where proteins, DNA, and viruses reside, and many other interesting structures for nanoscale applications. But we use DNA as a main material for this presentation, and thus it is most interesting. And I like to begin the presentation with a small scenario that motivates the topic. We imagine that there are many people in the future that suffer from multiple ailments. It is not uncommon that patients have multiple diseases and often have to spend a lot of time in stationary treatment when they get older. As an alternative, we want to inject people with nanonetworks that constantly monitor the health parameters of a person and transmits the results to a remote location, for example, a doctor that makes a judgment call about the patient's health. And in detail, the nanonetwork we inject might look as follows. There are certain kinds of nano devices that are pre-programmed to detect certain kinds of environmental parameters or disease markers. Once a detection takes place, those nano devices release their payload, which might be the building blocks for a so-called message molecule. This message molecule can only fully assemble once all the necessary building blocks are present, and it can compute a mathematical operation in the assembly process. And only when those assembly process has successfully finished will a message molecule with receptors at the top and the bottom form and now be present in the communication channel. Those molecules, like any other form of molecular communication, can be detected by other devices, and those can react upon the detection of the message molecule and, for example, release medical payload or release a fluorescent marker for further processing. Or it can simply communicate the measurement result to an external device. And the building block for those message molecules are so-called DNA tiles. You can create them from other materials too, but for the sake of already available materials, we focus on DNA tiles. They can be created from 4 DNA strands that are intertwined in a specific way. The strand at the top binds partially with the strand to the right, and the strand to the left binds partially with the strands to the bottom right. And the inherent binding property of DNA indicates that only the base pairings adenine and thymine can form a stable binding, and the base pairings of guanine and cytosine can also form a stable binding with each other. Thus, you can create a binding condition at the open ends of those DNA tiles in all cardinal directions. At the top, for example, there are open base pairings T and C, and they can form a binding with another tile with appropriate bindings of A and G. And lots of nanostructures can already be produced in the laboratory. What we can see here is a whole variety from 2015, and now we have already 6 years of more advancement in the areas. And it has already been possible to manufacture all kinds of shapes and an object at the nanoscale, like smiley faces, or even a DNA box with an open and closable lid, as you can see in this row here. And now to a formal definition of DNA tiles to properly understand the further content of the presentation. Mathematically speaking, DNA tiles are non-rotatable blocks with glues on all sides, possibly all sides. Those glues are indicated by black boxes, and the number of black boxes indicates the strength of the glue. In DNA language, this would be the length of the open DNA strand, for example, that can encode the strength of the binding. More fitting base pairings means a stronger binding. Each of those glues can further encode a label, and only glues with a fitting label can form stable binding. Again, this label encodes a fitting base pairing, the inverse base pairing strand that other tiles use to form stable bindings in DNA. And in the middle we have a marker, and this marker is mainly interesting for interpreting the results of computations or for seeing with fluorescence markers if something has happened, and for easier detection via atomic force microscopes. And those tile systems have some interesting properties. They can compute arbitrary functions from temperature 2 onwards. Temperature 2 just means that the tiles need a glue of at least strength 2 to form stable bindings with each other. And they are Turing complete, namely they can compute any mathematical function. Further, those tiles can construct and they can communicate in networks using the message molecule approach. And they can already obviously also create any other shape that you desire, like a box that contains medication. And it is, as mentioned, influenced by the temperature tau. And now to a small example. The process of such an assembly usually begins with a seed tile sigma. Without such a seed tile, nothing happens. Once the seed tile is introduced to a system, other tiles can form a stable binding with this seed tile. In this case, it is a temperature 2 system, so we need another fitting tile with a fitting label and a fitting glue strength of at least 2 at the bottom to form a stable binding. Now we have a similar situation, and at the left we have a glue with a label B of strength 2, and another tile can form a stable binding too. And now we have a more interesting situation, because the a total glue requirement of 2 can also be supplied from 2 different tiles, thus encoding a condition. This tile here with the marker 1 can form a stable binding here with 1 glue to the right with the tile sigma and 1 glue to the bottom with the label 1. And using the DNA example, the whole process might look as follows. We have lots of DNA tiles that are represented by the wiggly structures at the bottom, and the new tile to the top left can start to form a stable binding with the already existing assembly because the base pairings TAGC and GCCG are fitting with the other glues. Thus, binding can take place and a stable assembly can form. And what we can see here are the basic components that we use to encapsulate medication, for example, or tiles. It is a simple DNA box that can be opened via a DNA strand. The bottom part of the box binds relatively weakly with the lid, and once a fitting DNA marker sequence, for example, that indicates a disease, swims by, it forms a more stable binding, and the box opens and releases its payload, for example, the tiles or the medication. And now to a more appropriate example, the computation of a 4-bit AND message molecule that simulates a distributed consensus upon the detection of a disease. The process starts again with a seed tile sigma. And now we have 3 possibilities. At the top, left, and bottom, there can be tiles attached. At the top, there is a tile T that broadens the message molecule. At the bottom, a tile B that also broadens the message molecule in 2 dimensions. And in the middle, the first conditional tile, the first input in red, of the computation of the 4-bit AND. A 4-bit AND can only, in circuit language, propagate the signal if all inputs are 1. And in this case, all 4 conditional tiles have to be present in order for the message molecule to fully assemble, thus correctly representing the logical behavior of the logical AND. And now the message molecule can continue to assemble and can only continue, the border structure can only continue when the next conditional tile is present, and the process repeats until the very last conditional tile is present, and now a receptor that communicates the result of a computation can bind to the message molecule. And it is very important that the message molecule can only finish when the computation has been successfully terminated, only when tiles 1 to 4 are present. And as a network architecture, the whole process looks as follows. Again, we have nanosensors 1 to 4 that each discover a different release a different type of tile, but discover or measure the same disease parameter. Once measured, they release their 4 different tiles, 1, 2, 3, 4, and a message molecule that computes a distributed consensus among all those devices is computed. When all 4 tiles are present, all 4 devices or nanosensors agree, yes, we have detected a disease, and there's a very slight probability of error left. And when the message molecules have been assembled, they can be detected by nanobots, and they can react by releasing medication. And now we are very sure that there's really a disease. But we can do more. We can do a very general procedure that can compute any Boolean function. And for this, we take a computational problem and transform it into a Boolean formula if that is possible. It's not possible for all, but it's possible for many. Then we create a truth table from that Boolean formula and compute the disjunct normal form phi. And then we can create a message molecule for each of the clauses. And the Boolean formula is successfully evaluated once a single message molecule is successfully assembled, thus representing the behavior of the disjunct normal form. It is enough if a single clause, a single line of the truth table evaluates to 1. So now to a small example. Here we have a very general truth table for formula phi. And for every line of the truth table that has a 1 at the end, we create one message molecule that represents the truth values of all the literals, phi 1 to phi 2, etc. And at the right part, we see an example and the tiles that we see in the middle. And essentially, those are representations of the very same end message molecule that follow the very same principle, just the molecule is just flipped. And now, the entire message molecule can only fully assemble when all the literals, all the tiles that represent the literals, with the corresponding correct truth values are present. This is very important. And once a single of those message molecules is successfully finished, then we know that that Boolean formula evaluates to true. We don't have to wait for all the other stuff to assemble. A single message molecule that has been fully assembled is sufficient. And now we have a look at the entire process as a DNA-based nanonetwork. Again, we have a lot more, possibly, nanosensors now that detect different kinds of disease markers, release their tiles, and now in the medium there are all kinds of other tiles, and the message molecules, no matter what type, can only fully assemble when at least the building blocks for one of those message molecules are all present, and then a nanobot can detect and react upon the detection of the message message molecule. Yes. And now we have a whole array of problems that have been previously unclear of how to solve that can be solved with this approach. Namely, all the problems of the classes AC0 and NC1 can be solved. And they have been proposed as a material or as functions that are necessary for certain tasks in medical scenarios. And in those classes AC0 and NC1 are all kinds of additional problems that we can also solve. And from those problems, we can create even more complex programs for more complex problems. And the entire approach has less overhead than Turing-based style systems, has fewer errors, and is realistically possible to build at the nanoscale using already available materials. And in summary, what have we achieved? We have used DNA and found DNA as a suitable material for construction, computation, and communication to create fully functional nanonetworks at the nanoscale that are technically already possible to create. Second, it is possible to create those nanonetworks just using one building block, namely DNA tiles or DNA in general. And it is possible to compute any Boolean formula at the nanoscale via message molecules, via molecular communication systems with a self-assembling component. And DNA-based nanonetworks only rely on already available technologies and can be used to perform complex and arbitrary computations. And as of right now, we can't really create nanonetworks in the laboratory. There are already great approaches that use other materials by further miniaturization of circuit technologies. But this technology can really be created right now if someone wants to. I don't have the necessary expertise, but it is possible. Okay, now my presentation is over. Thank you very much for your attention. If there are any questions, now is a great time for it.